bones, joints, and soft tissue tumors. The adult human skeleton is composed of 206 bones that account for approximately 12% of body weight. Bone functions include mechanical support, force transmission, internal organ protection, and mineral homeostasis, and it also serves the extracellular component of bone, the matrix, is composed of osteoid 35% and minerals 65%. The latter, 1,171. 1,172. Its hardness and serves as a repository for 99% of the calcium. Its hardness and serves primarily hydroxyapatite, K2O4602. Give bone its hardness and serves as a repository for 99% of the calcium and 85% of the phosphorus in the body. Osteoid consists of type 1 collagen and smaller amounts of glycosaminoglycans and other proteins. One of these proteins, osteopontin, also called osteocalcin, is produced by osteoblasts and contributes to the regulation of bone formation, mineralization, and calcium homeostasis. Serum osteopontin levels are used as a specific marker of osteoblast activity. Bone maturation and metabolism are also sensitive to cytokines and growth factors and are thus regulated by diverse inputs, which include locally and systemically produced factors, as well as mechanical force. Bone matrix can be woven or lamellar figure 26. 1. Woven bone is produced rapidly, e.g. during fetal development or fracture repair, but the haphazard arrangement of collagen fibers imparts less structural integrity than the parallel collagen fibers of lamellar bone. Woven bone is always abnormal in adults, but its presence is not specific for any particular disease. Long bones are composed of a dense outer cortex and a central medulla. The latter is supported by bony trabeculi interspersed with marrow, which may be fatty white or hematopoietic red. Cells. The cellular components of bone include osteoblasts, osteocytes, and osteoclasts. Osteoblasts on the surface of the osteoid matrix synthesize, transport, and assemble matrix and regulate mineralization, figure 26-2A. Osteoblast activity is tightly regulated by hormonal and local mediators. Quiescent osteoblasts, which can be recognized by a decrease in cytoplasmic volume, may remain on the trabecular surface or become embedded within the matrix as osteocytes. Osteocytes are interconnected by an intricate network of dendritic cytoplasmic processes through tunnels canaliculi within the matrix. Osteocytes help control calcium and phosphate levels in the microenvironment, detect mechanical forces, and translate those forces into biologic activity, a process called mechanotransduction. Figure 26.2A, active osteoblasts synthesizing bone matrix. The surrounding spindle cells represent osteoprogenitor cells. B, 2 osteoclasts resorbing bone. Osteoclasts are specialized multinucleated macrophages that are derived from circulating monocytes and resorb bone, figure 26. 2B, surface integrin proteins allow osteoclasts to attach to the matrix and create a sealed extracellular trench resorption pit. Secretion of acid and neutral proteases, predominantly matrix metalloproteases, MMPs, into the pit results in dissolution of inorganic and organic bone components. Development. Most bones that form during embryogenesis develop from a cartilage mold via endochondral ossification. The cartilage mold anlagen is synthesized by mesenchymal precursor cells. A central medullary canal within the anlagen is created by chondroblasts at approximately 8 weeks of gestation. Simultaneously, osteoblasts begin to deposit the cortex beneath the nascent periosteum of the midshaft diaphysis. This forms a primary center of ossification, resulting in radial bone growth. At the longitudinal ends, epiphysis, endochondral ossification forms secondary centers of ossification. Eventually, plates of cartilage anlage become entrapped between the expanding centers of ossification, forming physes, or growth plates. Figure 26.3. Chondrocytes within the growth plates undergo sequential proliferation, hypertrophy, and apoptosis. Matrix mineralizes during apoptosis and is invaded by capillaries, providing the nutrients for activation of osteoblasts and osteoid synthesis. Most calcified cartilage matrices ultimately resorb, leaving only strut-shaped remnants that serve as scaffolding for bone deposits known as primary spongiosa, the earliest bone trabeculae. See figure 26. 3. Over time, this process produces longitudinal bone growth. Flat bones, for example the cranium, are formed by intramembranous ossification, in which a dense layer of mesenchyme is directly ossified by osteoblasts without a cartilage anlagen. Bones enlarge by deposition of new bone on a pre-existing surface, a process called appositional growth. Figure 26.3, active growth plate with ongoing endochondral ossification. 1, reserve zone. 2, zone of proliferation. 3, zone of hypertrophy. 4, zone of mineralization. 5, primary spongiosa. Local and systemic factors that regulate bone development include the following. Growth hormone GH, secreted by the anterior pituitary gland, induces and maintains chondrocyte proliferation. Thyroid hormone, secreted by the thyroid gland, acts on proliferating chondrocytes to induce hypertrophy. Indian hedgehog IgE is secreted locally by prehypertrophic chondrocytes and coordinates chondrocyte proliferation and differentiation with osteoblast proliferation. Parathyroid hormone-related protein PARP, produced by perichondral stromal cells and early proliferating chondrocytes, activates the PTH receptor to maintain chondrocyte proliferation. WNT growth factors are expressed in the growth plate, proliferating zone, and via frizzled and LRP5 receptors activate catenin to promote chondrocyte proliferation and maturation. SOX9 is a transcription factor expressed by proliferating, but not hypertrophic, chondrocytes that is essential for differentiation of chondrocyte precursors. RUNX2 is a transcription factor expressed in early hypertrophic chondrocytes and immature mesenchymal cells that controls terminal chondrocyte and osteoblast differentiation. Fibroblast growth factors, FGFs, are secreted by a variety of mesenchymal cells. FGFs, most notably FGF3, act on hypertrophic chondrocytes to inhibit proliferation and promote differentiation. Bone morphogenic proteins, BMPs, members of the TGF-beta family, are expressed at various stages of chondrocyte development and have diverse effects on chondrocyte proliferation and hypertrophy at the growth plate. Homeostasis and remodeling. The adult skeleton appears static but actually undergoes continuous change via a tightly regulated process known as remodeling. This process, which turns over approximately 10% of the skeleton each year, repairs damage and may change the shape of bones in response to mechanical forces. Remodeling occurs within the bone or basic multicellular unit BMU, which consists of a unit of coupled osteoblast and osteoclast activity on the bone surface. Osteoclast attachment, bone resorption, osteoblast attachment and proliferation, and finally, matrix synthesis occurs sequentially at the BMU. Events at the BMU are regulated by cell-to-cell interactions and cytokines and several signaling pathways, figure 26.4. One pathway involves 3 factors. 1, the transmembrane receptor activator for NFB rank, which is expressed on osteoclast precursors. 2, rank ligand rankle, which is expressed on osteoblasts and marrow stromal cells. And 3, osteoprotegerin OPG, a secreted decoy receptor made by osteoblasts and several other types of cells that binds rankle and thus prevents its interaction with rank. When stimulated by rankle, rank signaling activates NF-kappa, which is essential for the generation and survival of osteoclasts. A second important pathway involves macrophage colony stimulating factor MCSF, a factor produced by osteoblasts that is also crucial for the generation of osteoclasts. Finally, WNT proteins produced by osteoprogenitor cells bind to LRP5 and LRP6 on osteoblasts to activate beta-catenin signaling and osteoprotegerin OPG synthesis, figure 26. 1,174, chapter 26, bones, joints, and soft tissue tumors, figure 26.4, paracrine molecular mechanisms that regulate osteoclast formation and function. Osteoclasts are derived from the same mononuclear cells that differentiate into macrophages. Osteoblast, stromal cell membrane associated rankle binds to its receptor rank located on the cell surface of osteoclast precursors, functions in concert with macrophage colony stimulating factor MCSF. Rank activation induces precursor cells to become functional osteoclasts. Osteoblasts and stromal cells also secrete osteoprotegerin OPG, which acts as a decoy receptor for rankle, preventing it from binding the rank on osteoclast precursors. Consequently, OPG prevents bone resorption by inhibiting osteoclast differentiation. Conversely, osteocytes produce sclerostin, which inhibits WNT beta-catenin signaling and promotes bone formation. The importance of these pathways is emphasized by rare germline mutations in OPG, rank, rankle, LRP5, and sclerostin genes, which severely disrupt bone metabolism and produce congenital bone disorders described later. The balance between bone formation and resorption is modulated by rank and WNT signaling. For example, because OPG and rankle oppose one another, bone resorption or formation can be favored by increasing or decreasing the rank to OPG ratio, respectively. Systemic factors affecting this balance include hormones, parathyroid hormone, estrogen, testosterone, and glucocorticoids, vitamin D, inflammatory cytokines, e.g. IL-1, and growth factors, e.g. bone morphogenetic factors. Each of these acts by altering rank NF-kappa and WNT beta-catenin signaling. Parathyroid hormone, IL-1, and glucocorticoids promote osteoclast differentiation and bone turnover, while bone morphogenic proteins and sex hormones generally block osteoclast differentiation or activity by promoting OPG expression, alterations that favor bone deposition. Another level of control involves paracrine signaling between osteoblasts and osteoclasts. Matrix breakdown by osteoclast liberates and activates growth factors, cytokines, and enzymes, e.g. collagenase, some of which stimulate osteoblasts. Thus, substances that initiate bone deposition are released into the microenvironment during bone resorption. See Figure 26.5. Peak bone mass is achieved in early adulthood after the cessation of skeletal growth and is determined by factors that include vitamin D receptor and LRP, 5-6, polymorphisms, nutrition, physical activity, age, and hormonal status. Beginning in the fourth decade, however, resorption exceeds formation, resulting in a steady decline in skeletal mass. Developmental disorders of bone and cartilage. Developmental abnormalities of the skeleton often stem from inherited mutations and become apparent during the earliest stages of bone formation. In contrast, acquired diseases usually appear in adulthood. The spectrum of disorders of bone development is broad, and there is no standard approach to their classification. Here we will categorize the major diseases according to their pathogenesis. Developmental anomalies can result from localized disruption of the migration and condensation of mesenchyme dysostosis, or global disorganization of bone and/or cartilage dysplasia. Dysostosis may occur in isolation or as part of more complex syndromes, and are caused by defects in mesenchymal condensation and differentiation into cartilage analogs. The most common forms include complete absence of a bone or entire digit aplasia, extra bones or digits, supernumerary digit, and abnormal fusion of bones, e.g. syndactyly, craniosynostosis, genetic alterations that affect genes encoding transcription factors, especially homeobox genes, cytokines, and cytokine receptors, are especially common among dysostosis. In contrast, dysplasias arise from mutations in genes that control development or remodeling of the entire skeleton. It is important to note that while the term dysplasia in this context implies abnormal growth, it is not a precursor of neoplasia, as is the case with dysplasias of epithelial cells, Chapter 7. More than 350 skeletal dysostoses and dysplasias are recognized, most of which are extremely rare. The classification has evolved from clinical and radiographic descriptions to one that includes the causative genetic defects. Table 26.1 lists some of the better characterized developmental abnormalities and their associated defective genes. The relationships between specific mutations and phenotypes are complex. Different point mutations in a single gene, e.g. KAL2A1, can result in distinct phenotypes, while mutations in disparate genes, e.g. LRP5, RANKL, can lead to similar phenotypes. Defects in nuclear proteins and transcription factors. Defects in nuclear proteins and transcription factors, especially homeobox proteins, result in disorganized mesenchymal condensation and differentiation of osteoblasts and chondrocytes, leading to abnormal bone development. Figure 26.5, bone cells and their interrelated activities. Hormones, cytokines, growth factors, and signal transducing molecules are instrumental in bone formation and maturation, and allow communication between osteoblasts and osteoclasts. Bone resorption and formation and remodeling are coupled processes controlled by systemic factors and local cytokines, some of which are deposited in the bone matrix. BMP, bone morphogenic protein, LRP5-6, LDL receptor-related proteins 5 and 6. Brachydactyly types D and E are caused by mutations in the homeobox HOX13 gene and are characterized by shortening of the terminal phalanges of the thumb and big toe, respectively. Loss of function mutations in RUNX2 result in cleidocranial dysplasia, an autosomal dominant disorder characterized by patent fontanelles, delayed closure of cranial sutures, Wormian bones, extra bones that occur within a cranial suture, delayed eruption of secondary teeth, primitive clavicles, and short stature. Defects in hormones and signal transduction proteins. Achondroplasia is the most common skeletal dysplasia and a major cause of dwarfism. It is an autosomal dominant disorder caused by gain-of-function mutations in the FGF receptor 3, FGFR3 gene, approximately 90% of which stem from new mutations in the paternal allele. FGF-mediated FGFR3 activation normally inhibits endochondral growth. This effect is exaggerated by FGFR3 gain-of-function mutations. The retarded cartilage growth results in shortened proximal extremities, an enlarged head with bulging forehead, and depression of the root of the nose despite a trunk of relatively normal length. These skeletal abnormalities are not usually associated with changes in longevity, intelligence, or reproductive status. Thanatophoric dysplasia is the most common lethal form of dwarfism. It affects approximately 1 in every 20,000 live births and is caused by FGFR3 gain-of-function mutations distinct from those that cause achondroplasia, which appear to cause greater increases in FGFR3 signaling than those that produce achondroplasia, and thus a more severe phenotype. Affected individuals have disproportionately short micromelik limbs, frontal bossing, relative macrocephaly, a small chest cavity, and a bell-shaped abdomen. The underdeveloped thoracic cavity leads to respiratory insufficiency, and these individuals frequently die at birth or soon after. Histologic examination of the growth plate reveals reduced chondrocyte proliferation and disorganization within the zone of proliferation, defects in extracellular structural proteins, mutations in the major bone and cartilage collagens, types I, 2, I, and she give rise to highly variable presentations ranging from lethal disease to premature osteoarthritis, OA, type 1 collagen diseases, osteogenesis imperfecta. Osteogenesis imperfecta OI, or brittle bone disease, the most common inherited disorder of connective tissue, is a phenotypically heterogeneous disorder caused by deficiencies in type 1 collagen synthesis. OI principally affects bone, but it also impacts other tissues rich in type 1 collagen, joints, eyes, ears, skin, and teeth. It is caused by mutations in genes encoding the alpha-1 and alpha-2 chains of type 1 collagen. Many of the over 800 mutations identified lead to replacement of a glycine residue within the triple helical domain with another amino acid. Collagen synthesis and extracellular transport require triple helix formation, and these mutations result in misfolding of collagen polypeptides and defective assembly of higher-order collagen chains. Mutant collagens also interfere with assembly of wild-type collagen chains. That is, they exert a dominant negative effect, thereby explaining the autosomal dominant inheritance pattern. 1176 chapter. Table 26.1 Diseases of the Skeleton with Identified Genetic Defects. Modified from Munlos S. Olson VR. Heritable Diseases of the Skeleton. Part 1. Molecular Insights into Skeletal Development. Transcription. The fundamental abnormality in OI is too little bone, resulting in extreme skeletal fragility. Other findings include blue sclerae caused by decreased collagen content, making the sclera translucent and allowing partial visualization of the underlying choroid, hearing loss related to both the sensory neural deficit and impeded conduction due to abnormalities in the bones of the middle, and inner ear dental imperfection, small, misshapen, and blue-yellow teeth, secondary to dentin deficiency. OI is separated into 4 major clinical subtypes of varying severity, table 26.2. Mutations that result in decreased synthesis of qualitatively normal collagen are associated with mild skeletal abnormalities. More severe or lethal phenotypes are associated with mutant collagens that interfere with triple helix formation. The type 2 variant, which is uniformly fatal in utero or during the perinatal period, is characterized by extraordinary bone fragility and multiple intrauterine fractures, figure 26. In contrast, individuals with type 1 OI have a normal lifespan, but experience childhood fractures that decrease in frequency following puberty. Defects in metabolic pathways, enzymes, ion channels, and transporters. Osteopetrosis. Osteopetrosis comprises a group of rare genetic diseases characterized by reduced bone resorption due to deficient osteoclast development or function, which leads to diffuse symmetric skeletal sclerosis. Although the term osteopetrosis implies that the bones are stone-like, they are actually brittle and fracture easily. Osteopetrosis is classified into variants based on both the mode of inheritance and the severity of clinical findings. Pathogenesis. Most of the mutations underlying osteopetrosis interfere with acidification of the osteoclast resorption pit, which is required for the dissolution of calcium hydroxyapatite within the matrix. For example, Albers-Schomburg disease, a mild autosomal dominant form of osteopetrosis, is caused by mutation of CLCN7, which encodes a proton chloride. Bone 1177, table 26. Exchanger on the osteoclast surface that is required for resorption pit acidification. Similarly, most cases of autosomal recessive osteopetrosis are caused by a mutation of TCRG1, which encodes a subunit of the osteoclast vacuolar H+ aptase that is also necessary for acidification of the resorption pit. Other causes of autosomal recessive osteopetrosis include defects in carbonic anhydrase 2CA2, which, like all carbonic anhydrase isozymes, generates protons and bicarbonate from carbon dioxide and water. CA2 facilitates resorption pit acidification by osteoclasts and urinary acidification by renal tubular epithelial cells. Osteopetrosis due to CA2 mutations is therefore accompanied by renal tubular acidosis. Mutations in IKBKG, which encodes NEMO, the regulatory subunit of the inhibitor of CAPEB kinase IKK complex that is involved in NF-CAPEB activation, is a cause of osteopetrosis that is not due to defective acidification. NEMO, which is X-linked, is required for osteoclastogenesis and osteoclast survival, which are regulated by NF-kappa. Because NF-kappa serves many other functions, IKBKG mutations result in a multisystem disorder called X-linked anhydrotic ectodermal dysplasia with immunodeficiency that includes osteopetrosis and morphology. Due to deficient osteoclast activity, bones involved by osteopetrosis lack a medullary canal. The ends of long bones are bulbous Erlenmeyer flash deformity and misshapen figure 26-7. The neuroforamina are small and can compress exiting nerves. The primary spongiosa, which is normally removed during growth, persists and fills the medullary cavity, leaving no room for the hematopoietic marrow, and preventing the formation of mature trabeculi, figure 26-8.